One of the things that Nathan mentioned and that we showed a short video of was the use of these barcoded colonies, colonies where every bee is individually marked. And here we want to show you some video of inside the bee lab, how these bees are prepared and how the colonies are then used. So here we can see a video of a student assistant worker inside the bee lab in his workspace. He's got a clear tub full of day-old bees that have already been marked, and he's also got a preparatory tub that has a little bit of ice in it. Here's someone else doing the same procedure. Bees are chilled on ice as day-olds, which I'll talk a little bit more about later. They're chilled until they don't move anymore, and they can be very carefully handled. They're then lined up while chilled, and a very small code tag is glued onto the bee's thorax. And then they're very carefully kept in a container until they're placed into the colony. Here we can see the application of one of the tags. The bee is held very carefully. A tiny dab of super glue is added onto the bee's back. And then very carefully, with great care taken to orient the tag properly, the little tag is glued onto her back. These bees can then be put into a colony and they act totally normally, but both a human observer and a computer system hooked to a camera can now observe and track all the bees. The computer can do this at a remarkable accuracy and provide us with an enormous amount of behavioral data to use in a variety of experiments.
My name is Ashley St. Clair, and I'm a postdoctoral research associate in the Dolezal lab at University of Illinois. My research here is focusing on investigating honeybee queen health and how environmental stressors impact her health, specifically looking at her egg-laying success and viability of those eggs. So what do I mean by environmental stressors? Well, the honeybee queen is never really leaving the colony except for her mating flight. However, the foragers in the colony are going to go out and they may come in contact with many different things in the environment while they're out. So some of these can be while they're foraging for resources, they may find, depending on the landscape, reduced nutritional quality of available resources or just overall reduced abundance of resources. Some other things they may contact are toxins in the environment like pesticides. They can come in contact with pathogens, which can be spread throughout the colony, along with a lot of other things. And so, like I said, the queen is never really leaving the colony, so it can be really difficult to quantify how these external exposures can impact the queen. There's a lot of different ways that she may be exposed to this through what the foragers bring back. It could be through direct contact from another worker that's been exposed. It could be through feeding interactions along with a lot of other things. So this can be really hard to control in a full-scale colony because we don't really know what the foragers are coming in contact with or exactly how the queen may be getting exposed to those things. So something we've developed as an alternative way to investigate how queen health and her egg-laying success is impacted by these stressors is by taking this to a very small cage system that we can use in the lab. This provides a really controlled system where we can quantify these things and directly observe the effects on the queen without having all the complicated interactions that are in the full-scale colony. Because we have this cage system in the lab, we need to very carefully control the environment so that it can be as similar to the honeybee colony that would be out in the natural outside. So what we have are these incubators that we can control the temperature as well as the humidity to mimic the environmental conditions as best as possible. We can also control the light in here if we wanted to. So these are the cages that I have. So each of these little cages has some workers, about 60 workers in there, and a queen. And we can actually feed them different, you can have lots of different experimental procedures, but right now I'm feeding them different quality of pollens to investigate how pollen nutrition impacts queen egg laying. So we have honey here, some sucrose. We also have some water that we provide them on the other side, and then this is the pollen. The great thing about these cages is that they have some excluder material here at the bottom so that the queens cannot get down here and access the food. That way we can control exactly how the queen is exposed, and this has to be through feeding interactions. So as you can see in this cage, it looks like the queen has actually been laying some eggs. Let's see if I can get a close-up here so you can see them. Here we go. So each one of these individual cells, the queen has been laying eggs in there. So we will actually pull this this plate with the cells out, and we can individually count each egg so that we can quantify what her success is like with the different treatments. And so here's an example of what I'm talking about when I say pollen treatment. So I have two different pollens here. This is a rock rose pollen that we know has a high nutritional quality and results in high queen egg laying. And this is another experimental pollen that we're testing out in the lab right now, which is a polyfloral mix from Arizona. And so I'm running a trial to see how well this mix compares to the rock rose that we know induces good queen egg laying, so that we can see what the nutritional quality of this Arizona pollen is. So with these cages, they're actually really easy to use. You can just pull out this outer cover here that keeps the frame in place, and then you can place it on the inside. Very gently, you can just kind of knock the bees off. We don't want to injure any bees. And we'll just slide this down in there. And then we can pull this plate out. And we can actually look straight down in there and start to see all those eggs that the queen has laid. This cage is actually really great as well. I'm not doing it for this experiment, but if we wanted to, we could observe her in the incubators or in a walk-in chamber with the interactions that the bees are having. So if we wanted to see not only how different treatments affected her egg laying success, but also the interactions between her and the workers, we could just see the queen. So there she is right here. She's the larger bodied bee. Nice big abdomen. I mean, you can see some actually right now, some other bees kind of tending to her, grooming her. So they call that retinue. And the bees will kind of court her consistently throughout. So this would provide a really great opportunity in a controlled way with little disturbance to the queen to actually observe those interactions. So I went ahead and counted eggs in this plate, and I found 129 single eggs in a cell, and then twice she laid 2 eggs in a single cell. And so now that I have counted that, I've actually cleaned the eggs out of the frame so I can put those back into the colony. If I were going to go ahead and look at egg viability, I can actually just leave this plate in the incubator and the eggs would hatch out. And then I could come back in about 3 days and I could just see how many of them hatched to get a percentage of eggs that were successful. These frames are really cool because they are sort of modified from what a 96 well plate looks like, but they are actually the standard size of a honeycomb cell. And so not only will the queen lay eggs in there, and sometimes you can see they will put a little honey or something in there if they have the resources to do that. But you can actually place this inside a full colony, and the bees will rear out the pupae in there and cap them, and possibly even they could develop into adult bees, although I've never done that myself. But for this experiment, I'm just going to place this back inside the cage, and now I can pull that inner cover back out and place that back on the outside to keep it in place. And now the queen can begin laying in this frame again. So I'll come back every single day and I will check this and clean it out so that she can have a fresh space to lay every day. And now that I'm done with that, what I need to do is just replace her pollen treatment. So I'm going through every day and I'm providing them fresh pollen with the different experimental treatments. So in order to do that, I actually can just remove this old pollen from inside there. You just want to make sure that there's no bees actively foraging in there, which it looks like there is one right now. So we'll kind of coax her back. Okay, so I've gotten the pollen tube out, and I've just replaced it with an empty tube right now just to plug the hole so that the workers don't climb out of the cage. And what I'm going to do next is take our other tube. So we've cut off the end of it. That way we can just plug pollen right inside there and let the bees get to it. And I'm just going to fill it up with some of this fresh pollen. So this is granulated pollen that was collected by bees, honeybees, and then we ground it up and mix it with a little bit of honey, which is why it has this sort of sticky texture to it. And this just makes it a little easier for the bees to consume it from this tube. And now we can take this old frame out of there and put the new fresh pollen tube back in. There we go. All right, now they have some fresh food in there. So we can just place this back inside the incubator. And we will come back tomorrow and see how many more eggs she's laid.
So one of the tools that we use a lot in my lab are these bee cages. So these are these little plexiglass cages that we can use to house a relatively small number of bees and do lots of different experiments to see how they respond to pathogens or different diets or to pesticide exposure. And so here we can see a short video of Lincoln Taylor, a student working in the lab, making up these cages at the bee lab. And so he's doing it in one way, which is where you can very quickly count bees out. And so you can count to 30 or 35 or 50 bees, count them into a plastic container like the Tupperware he's throwing them into now, and then you can then dump them in. Another way, if we don't care so much about the exact number, is just to weigh them out. And so we use the estimate of about 0.1 grams per bee. You can just weigh a container and then kind of estimate the number of bees. But here he's counted them all out, and he's now knocking them down to the bottom, and then just pour them right into that little cage right there. And you can see that they are fine in there. We do this only with newly emerged bees, day-olds, bees that are have emerged from a cap in less than 24 hours. These bees can't fly, they can't sting, and they're really docile. And so that's how it's very easy to do this kind of counting out and standardization. Also, we know that bees' biology is really affected by their age. Day-old bees are quite different than nurse bees, and those are very different than foragers, both in their behavior, their physiology, and how they respond to things like infections. And so this also gives us the ability to control these experiments very well, that we know all the bees across all our treatment groups are all the same age at the beginning of the experiment. If we were comparing nurses to foragers, we'd get very different results. Another thing we can do with this is very carefully control the source of the bees. So we can use bees that have emerged from just a single colony. Some people at the bee lab even use just a single colony where the queen was artificially inseminated to simulate a mating with just a single drone. And so she has very highly related worker offspring. Or we can look at different queen lines, or we can, if we're interested in things that aren't genetic, we can take bees from many different colonies, 10, 15 different colonies, and mix them all together. As day-olds, they don't care, and they're very happy to be mixed. And then we can create this like homogeneous average bee and put those into cages for other types of experiments. And here we can see what these cages look like once we start observing them. So here are some that have been placed in an incubator by Lincoln. And we can see the bees walking around in there. They're clear, they've got air holes, so they're easy to observe. In these ones, we can see in the bottom left of the cage a pollen ball to feed the bees. The little tube coming out the side is full of just sugar solution, so they can drink that. And then these experiments are being done to look at how workers may or may not transmit diseases to developing queens. And so what many of you will recognize in the top of these cages is a queen cell that we grafted queen larvae into, or a larvae into, put them in a starter. And then after they had started, we can then place those into a cage like so, and the workers inside will then care for that queen larva. And so you can see at this stage in the experiment, most of these, if not all of them, have been capped. And so the workers inside this cage, which we can either infect or not infect, take care of that larva and eventually cap it. So we can see that one looks like it might not yet be capped. And so this gives us the ability to control the environment very tightly for lots and lots of different types of experiments. And of course, this is not a beehive. These are not colonies, but they are a small group of bees, a small social group. And so we can observe a lot of things in these cages that are really useful, even though they're not exactly the same as the real colony. And so they let us control a lot of things you could never control in the field, and they give us an ability to have a really large sample size so we can kind of see what variation might exist. These can then be scaled up to field level or semi-field experiments to see how much is actually going on in the field.